So another flash quiz is upon us. They keep sneaking up on us. In what type of heart block do the atria and the ventricles beat independently of each other? We know this. Think about what would cause the atria to beat independently of the ventricles. Something would have to completely obliterate the electrical connection between the two. We discussed this, and we know this to be third degree heart block. So, we're going to talk about baroreceptors and chemoreceptors. Before we do that, let's do a couple of things. Let's actually discuss what exactly a neuronal baroreceptor reflex loop is composed of. It is composed of four things. Number one, you need something that is detecting changes in pressure or ion concentrations and changing this into an electrical signal. And these are actually our receptors. Baro, meaning pressure, and chemo, meaning ions. Number two, we need an afferent pathway to conduct it to the brain. In this particular section, that will be the glossopharyngeal and the vagus nerve. Number three, we need somewhere in the brain to put it all together. This is the solitary nucleus in the medulla for this particular reflex arch. And four, we need an efferent outflow, which will most likely be a autonomic component, either the sympathetic or parasympathetic. The receptors we are going to discuss first are the baroreceptors in the aortic arch and the carotid sinus. Now, why are they in these locations? So let's quickly draw a diagram that will help us visualize what exactly we're going to be talking about. So we have our heart, and then we have our aortic arch coming up. Now don't forget, you have your innominate or brachiocephalic giving off your right subclavian, and then giving off your right common carotid. And then you have the left common carotid, and then the left subclavian. The ones we're worried about are the left common carotid and the right common carotid, because we know that eventually they will split into the two different components, which are the internal carotids and the external carotids. So there's actually baroreceptors in the aortic arch, and these travel up both vagus nerves. So these are 10. And then we have receptors that are kind of wrapped around the vasculature at the bifurcation of the common carotid, where it bifurcates into the internal and the external carotids. And these send the afferent pathway up via the glossopharyngeal, which is nerve 9. So the aortic arch is a great place to tell you how the pressure is immediately after you eject blood out of the aorta. And the carotid sinus is immediately distal to a very important bifurcation of the internal and external carotids. You always have to make sure your brain is getting perfused. So these receptors are highly branched fibers that interlock with the elastic fibers of the vessel wall. So an increase in pressure will stretch that wall, and that means you stretch the fibers of that wall, and then you stretch the mechanoreceptors that are actually embedded within the elastic fibers of the aortic arch vessels and the carotid sinus that is sitting at the bifurcation of the common carotid into the internal and external carotid arteries. So when you stretch these receptors, you induce the receptor ion channels to undergo conformational changes to allow influx of ions. And then that gives us depolarization. There we go. That's what we're looking for. So the depolarization of the receptors are actually occurring at the afferent nerve terminals. So right where those mechanoreceptors are going to join the nerves that travel up to the medulla. And remember, these nerves are 9 and 10, the glossopharyngeal and the vagus. Now remember, the afferent pathway for the carotid sinus will travel via the glossopharyngeal, while the afferent signal for the aortic arch will travel via the vagus nerve. The left recurrent laryngeal, which is a branch of the vagus, loops under the aortic arch. So this is kind of a good way to remember that the baroreceptors that are located within the aortic arch will also be mediated by the afferent pathway, which is taken up to the medulla by the vagus nerve. The sensory fibers from the aortic arch actually join the superior laryngeal nerve, which is also a component of the vagus, to travel up to the brain. So this is another great way to remember the pathway. So now let's talk about the response. So the afferent fibers from the carotid sinus traveling via the glossopharyngeal nerve, and the afferent fibers from the aortic arch traveling via the vagus nerve eventually reach the solitary nucleus of the medulla. As you can see, we have two options. We can either downregulate the parasympathetic or upregulate the sympathetic nervous system. Let's incorporate the physiology of the carotid massage maneuver that we discussed earlier. Remember, this is an important maneuver because you can actually use it clinically, and I've seen it used many times, to depress someone's conduction through their AV node. Now, you can actually use this to simulate an increase in pressure. You usually perform it when someone has some sort of supraventricular tachycardia, and you want to decrease the conductions through the AV node so you can increase the diastolic filling period and also decrease the rate at which the ventricles are contracting. So when you massage on the carotid sinus, you're basically simulating an increase in the transmural pressure, which those baroreceptors sense. So that's basically like simulating an increase in blood pressure. That means the baroreceptors will be stretched, and eventually we have depolarization of the afferent nerve fibers to the medulla. Remember, in the carotid sinus, so that's occurring via the glossopharyngeal. So if we are simulating increased blood pressure by pressing on the carotid sinus, we will simulate an increase in blood pressure. So this diagram shows you that the solitary nucleus can control the outflow to the parasympathetic and the outflow to the sympathetic. The diagram is a little bit limited in the sense that you can actually increase or decrease either route. What is important to understand here is that we're simulating an increase in blood pressure. Our bodies do not like acute changes, whether up or down, in our blood pressure. So if we're simulating increased blood pressure by pressing on the carotid sinus, do you think we should activate the parasympathetic nervous system or the sympathetic nervous system? Well, we'll probably activate the parasympathetic nervous system and also downregulate our sympathetic outflow, because we need to decrease our blood pressure by changing the cardiac output or changing the systemic vascular resistance. Remember, blood pressure is cardiac output times the total peripheral resistance. So we need to modulate one of these two, or both. So some neurons in the solitary nucleus in the medulla project to the vasomotor region of the medulla because we want to decrease pressure. We inhibit the vasomotor region of the medulla, and that means we get a decrease in vasoconstriction. On the other hand, let's say we have hypotension, where we have a decrease in blood pressure. Then the solitary nucleus will activate the sympathetic nervous system outflow. You stimulate that vasomotor center and cause sympathetic outflow, such as release of epinephrine and norepinephrine, which stimulates alpha-1 receptors on arterioles, and you get arteriolar vasoconstriction, and you also get some venoconstriction, and that increases your blood pressure, as well as you get beta-1 stimulation that causes an increase in cyclic AMP and will increase the frequency, F is for frequency, at which you open the Fonny channels that are responsible for our Fonny current. And we also stimulate beta-1 receptors that increase cyclic AMP in contractile cells, which leads to an increase in intracellular calcium, causing an increased contractility. There are also projections of the solitary nucleus to the dorsal motor nucleus and the nucleus ambiguous of the vagus nerve. So let's say again that we're simulating increased transmural pressure on a vessel by massaging the carotid sinus. The response will be parasympathetic nervous system mediated, because these vagal nuclei will release acetylcholine on to the sinoatrial node and the AV node, which will stimulate the G inhibitory subunit of the G protein complex which leads to a decrease in cyclic AMP, which we know will limit the increase in intracellular calcium. And intracellular calcium is denoted by Ca2+ with an I for intracellular.